Hello everyone. In this video, we're going to learn about the principle and procedure of negative staining. The main purpose of negative staining is to study the morphological shape, size, and arrangement of the bacterial cells that is difficult to stain. For example, in bacteria like Spirilla. It can also be used to stain cells that are too delicate to be heat fixed. It is also used to prepare biological samples for electron microscopy. It is used to view viruses, bacteria, bacterial flagella, biological membrane structures, and proteins or protein aggregates, which all have a low electron scattering power. The principle of negative staining. Negative staining requires an acidic dye such as India ink or nigrosin. Indian ink or nigrosin is an acidic stain. This means that the stain readily gives up a hydrogen ion and the chromophore of the dye becomes negatively charged. Since the surface of most bacterial cells is negatively charged, the cell surface repels the stain. The glass of the slide will stain, but the bacterial cells will not. The bacteria will show up as clear spots against a dark background. So here we are starting with the procedure of negative staining. The reagent used here is nigrosin. So with the help of a dropper, we will put a drop of nigrosin at one end of the clean and well-flamed slide. The remaining nigrosin is put back in the bottle. Then the inoculating loop has to be properly sterilized until the wire is red heated. This is to avoid contamination. It is cooled down so that the organisms do not die while we pick it up. So a loopful of bacterial culture is picked up with the help of the inoculation loop, and it is then mixed well on the slide. We will sterilize it again and put the loop aside. For the next step, we will use another slide and place it against the drop of suspended organism at a 45-degree angle and spread it. And then allow the slide to dry, air dry. Once the smear is air dried, it is then observed under the microscope at an oil immersion 100x objective. In negative staining preparations, the bacterial cells observed as the clear transparent bodies or objects may be of variable size and shape against a dark background. So here we see rod-shaped bacteria against a dark background. So this is how the negative staining procedure is performed.